Hello everyone, welcome to the Bioinformatics Practical sessions. Today we will be discussing multiple sequence alignment with the Clustal W or Clustal Omega programs. Now, in the LMS platform, open the seventh practical item, that is a multiple sequence alignment with the Clustal W or Omega. The objective of this practical is to perform the multiple sequence alignment with the packages Clustal W or Clustal Omega. Now, you can obtain any FASTA file or multiple sequence containing FASTA file from NCBI, for which you require a BLAST algorithm, and for which you require NCBI BLAST program. So this is the BLAST program. Open a nucleotide BLAST. Now in the previous example, for example, you downloaded one of the sequences. I am copying one of the sequence, a nucleotide sequence. So you can paste the sequence here, or another option, you can paste as mentioned here, for example, the accession number, insulin gene accession number, that can be pasted here. So you can use the DNA sequence or accession number. So here you can copy the accession number. For every gene there will be an accession number, and that can be pasted on that pass. Now you can see that automatically the job title is marked here. You are looking for nucleotide collection, not the protein collection. Highly similar sequences with mega blast should be selected by default. Perform a blast. Now here is the result. Now select a few bases. A few result, like deselect all. The first one, the second one. The accession length is almost same, and this is a very long sequence. There is no point in aligning a larger sequence with a smaller one. So whereas this one is about almost the similar length, so you are selecting this one, and these are all shorter sequences, and we will look for another sequences with a similar length, like okay. So now I selected some of the sequences, but their similarity ranges from about 100% to 81% to 73%, etc. So 5 different sequences with the different similarity were selected. These sequences can be now downloaded in FASTA format. So for which you require download FASTA, not the complete sequence, align the sequence, save. So that will be saved as a sequence dump file in a Windows. So you can open this file, for example, in a notepad you can open. So all the 5 sequences were now downloaded in FASTA format. Okay. Now you come to the procedure. In the procedure, we have 2 websites given. One is for Clustal W and one is for Clustal Omega. So from the NCBA website, you performed a blast and downloaded 5 similar sequences, aligned sequences in FASTA format. Now open this tool, that is Clustal W tool, and see there is a search box. Now you can either browse and locate this sequence dump file, or you can paste the sequence here. Anyway, I am selecting and pasting the sequence, complete sequence, with the command line or the complete sequence. Okay, I am pasting all the sequences here one by one. This is including the command line. Usually it is not provided something like that, but now the entire sequence file is copied and pasted here. If you want, other parameters can be changed, but you can keep it as such. Execute multiple sequence alignment. See, alignment scores and the sequences were now aligned. The stars indicate high similarity. All the 5 strands is having the same sequence, whereas dots show some consensus range where majority of the bases were in that at that position will be similar, but some changes will be there. And if there is a gap, there is no star or dot available. So similar conserved domains, for example, this region of the concerned peptide is having a highly similar sequence. This is a conserved domain, maybe an active site. Whereas the other regions may not be relevant, or uh, it is not contributing to any significant structural part or biological function in this region. That's why it is not conserved. So conserved regions of the proteins can be easily determined by aligning the sequences and finding the similarities in that sequence. So the stars indicate high similarity. So this is how Clustal W results look like. Okay. And another one is Clustal Omega. You can open that one also. It will also provide you with another search box. Then select the parameter or output format. If you want, select the parameters or leave it as such and submit. It will also give you a similar result. All the 5 sequences were aligned. Similar regions were marked with stars. And you can click on colors, show colors, to show a colorful representation. Then phylogenetic tree construction is also possible. A cladogram of the 5 sequences were now created in the phylogenetic tree based on the alignment. The phylogenetic tree data can be downloaded from this button. This is the tree data, which can be used for plotting the tree using some other softwares. So this is about Clustal W and Clustal Omega, where we will perform multiple sequence alignment using this Clustal package, Clustal W, Clustal Omega. Okay.